Hello everyone. Today let us see about the vectors for yeast. Saccharomyces cerevisiae is an yeast which is the most important one in the biotechnology. So development of cloning vectors for yeast was initially stimulated by the discovery of a plasmid which is present in the most strains of Saccharomyces cerevisiae. So the 2-mu plasmid is a common plasmid which is found in the eukaryotic cells. Let us see about the characteristics of 2-mu-M plasmid. So it is an excellent basis for a cloning vector and it is covalently close to circular DNA molecule. So the size of this plasmid is 6 kilobase and the copy number is between 70 and 200 and it is located in the nucleus of yeast cell. The replication 1 and replication 2 sites are involved in the replication of the plasmid. Then FLP codes for a protein that can convert the A form of plasmid to the B form in which the gene order has been rearranged by the intramolecular recombination. And the D encodes recombination specific factors. Next, vectors based on the 2-mu-M plasmid and it consists of 5 different types. So first one is yeast episomal plasmid. So it was first constructed by Bex in 1978 by recombining an E. coli cloning vector with the naturally occurring yeast 2-mu-M plasmid, and the vectors derived from 2-mu-M plasmids are called as the episomal plasmids. So for example, YEP13. So this is the structure of YEP13. And the features of YEP13 are it can replicate as an independent plasmid and it is a shuttle vector. It contains 2-mu-M origin of replication and selectable leucine 2 gene and it includes the entire PBR322 sequence and it can replicate and be selected for in both yeast and E. coli. It has a copy number of 50 to 100 per haploid cell and it is easy to recover the recombinant DNA molecule from a transformed yeast colony. The second one is the yeast integrative plasmids. It is a bacterial plasmid which carries a yeast gene and this is the PBR322 plasmid with the uracil-3 gene instead. Uracil-3 gene is a selectable marker and it codes for the orotidine 5-phosphate decarboxylase and it does not contain 2-mu-M plasmid and cannot replicate as plasmids. And it has all features of yeast cloning vectors except the ORI site. And these plasmids therefore replicate only by integration into yeast chromosome and the copy number is 1 per cell and it produces very stable recombinants. So example for this plasmid is YIP5 and in this structure consists of amphotericin and tetracycline and this is the uracil-3 gene. The third one, it is yeast replicative plasmids. So it is constructed by Struhel et al. In 1979. So they isolated the chromosomal fragments of DNA which carry sequences that enable the E. coli vectors to replicate in yeast cells. So such sequences are known as the ARS, which is nothing but the autonomously replicating sequences and act as a origin of replication. It can able to multiply as independent plasmids and this plasmid is made up of PBR322 plus the yeast gene TRP1, that is the transient receptor potential, and this gene TRP1 is involved in the tryptophan biosynthesis and it is located adjacent to the chromosomal origin of replication. So the yeast DNA fragment present in YRP7 contains both the TRP1 and the origin of replication. So this is the structure of YRP7. Fourth one, yeast centromeric plasmids. So this plasmid is autonomously replicating plasmids and it considered as the low copy vectors and incorporate part of an autonomously replicating sequence along with the part of the centromere sequence. So and it is also a single copy just like the yeast integrative plasmids and the centromere sequence vectors are stable without the integration. And this plasmids transform yeast at high efficiency. So example for this yeast centromeric plasmid is YCP50 and this is the structure. The last one is the yeast artificial chromosome. So it is the genetically engineered chromosomes which is derived from the DNA of the yeast and it is a human engineered DNA molecule used to clone the DNA sequences in yeast cells. and they are the products of a recombinant DNA cloning methodology to isolate and propagate very large segments of DNA in a yeast host. By inserting large fragments of DNA, the inserted sequences can be cloned and physically mapped using a process called the chromosome walking. So the amount of DNA that can be cloned into a yeast artificial chromosome is on average from 200 to 500 kilobase, and however as much as 1 megabase can be cloned into a yeast artificial chromosome. And in this yeast artificial chromosome we can able to see the markers like A and B, then eco-R1 site, origin of replication, then centromere sequence and amphisilin. Working principles of YAC. It is an artificially constructed system that can undergo the replication process. So the design of YAC allows extremely large segments of genetic material to be inserted. Subsequent rounds of replication produce many copies of the inserted sequence in a genetic procedure known as the cloning. So the principle is similar to that for plasmids or cosmids. So the experimenter introduces some typical elements that are necessary for the correct replication. So in the case of YACs, the replication origins are the centromeres and telomeres of the yeast chromosomes, which must be inserted into the DNA being cloned. So the constructs can be transformed in yeast spheroplast and are then replicated there. In contrast to the vectors, YACs are not circular, they are made of linear DNA. So the process of YAC. So YAC vector is initially propagated as a circular plasmid inside bacterial host utilizing bacterial ovary site. The circular plasmid is cut at a specific site using restriction enzymes to generate a linear chromosome with 2 telomere sites at terminals. So the linear chromosome is again digested at a specific site with 2 arms with a different selection marker. So the genomic insert is then ligated into YAC vector using DNA ligase enzyme. So the recombinant vectors are transformed into yeast cells and screened for the selection markers to obtain recombinant colonies. Let us see about the advantages of yeast artificial chromosomes. So this YAC provides the largest insert capacity of any cloning system. Yeast expression vectors such as the yeast artificial chromosome, yeast integrating plasmids, yeast episomal plasmids have advantages over bacterial artificial chromosomes, that is the BAC, and they can be used to express eukaryotic proteins that require post-translational modification. So a major advantage of cloning in yeast eukaryote is that many sequences that are unstable, underrepresented or absent when cloned into prokaryotic systems remain stable and intact in YAC clones. It is possible to reintroduce YACs intact into mammalian cells where the introduced mammalian genes are expressed and used to study the functions of genes in the context of flanking sequences. Use of yeast artificial chromosomes. So YACs were originally constructed in order to study the chromosome behavior in mitosis and meiosis without the complications of manipulating and destabilizing native chromosomes. The YACs representing contiguous stretches of genomic DNA have provided a physical map framework for the human, mouse, and even Arabidopsis genomes. YACs are extremely popular for those trying to analyze the entire genomes. What are the limitations of yeast artificial chromosomes? A problem encountered in constructing and using YAC libraries is that they typically contain clones that are chimeric, that is, they contain DNA in a single clone from different locations in the genome. YAC clones frequently contain deletions, rearrangements, or non-contiguous pieces of the cloned DNA. As a result, each YAC clone must be carefully analyzed to be sure that no rearrangements of the DNA has occurred. So the efficiency of cloning is low. It is about 1000 clones are obtained per microgram of vector and insert DNA. Then YAC has been found to be less stable than BACs, that is the bacterial artificial chromosomes. The yield of YAC DNA isolated from a yeast clone containing a YAC is quite low. The YAC DNA is only a few percent of the total DNA in the recombinant yeast cell, and it is difficult to obtain even 1 μg of YAC DNA. The cloning of YACs is too complicated to be carried out by a lone researchers. Thank you.